So now that we've explored the vessels of the lymphatic system as well as the cells of the lymphatic system, let's take a deeper look at the various lymphatic tissues and lymphatic organs. Lymphatic or lymphoid tissue is aggregations of lymphocytes in connective tissues of the mucous membranes and various organs. The simplest form is diffuse lymphatic tissue. This is where lymphocytes are scattered rather than densely clustered, and they're particularly prevalent in body passages that open to the exterior environment. So that would include the respiratory tract, digestive tract, urinary, and reproductive tracts. In these tracts, it's called mucosa-associated lymphatic tissue, or MALT. In some places, the lymphocytes and macrophages congregate in more dense masses, so not diffuse but dense tissue. And they accumulate in these locations in response to invading pathogens. And they'll come and go as necessary. However, they're a very constant feature in the lymph nodes, the tonsils, and the appendix. And in the ileum and the distal portion of the small intestine, these dense clusters of lymphocytes and macrophages are called Peyer's patches. This makes lymphatic tissue highly available to the digestive system in case there are invading pathogens that come in through that route. Now, in contrast to diffuse lymphatic tissue and lymphatic nodules, lymphatic organs have well-defined anatomical sites, and they have connective tissue capsules that separate the lymphatic tissue from the neighboring tissues. The primary lymphatic organs we'll see are red bone marrow and the thymus. And they're considered primary because these are the sites where the T and B lymphocytes become immunocompetent. That is, they become able to recognize and respond to antigens. Antigens are the invaders. The secondary lymphatic organs are then populated by the T and B lymphocytes that have matured in either the bone or the thymus. These organs are the lymph nodes, tonsils, and the spleen. Let's first look at red bone marrow. As we've already learned, there are two types of red bone marrow. There's red and yellow. And the red bone marrow is very prevalent in all the long bones of children and less prevalent in adults. But red bone marrow is the type of marrow that's involved in hemopoiesis, or blood formation, as well as immunity, because as we know, all the white blood cells are involved in immune functions. So bone marrow is a soft, loosely organized, and highly vascular material. There's an image of it over here on the right. It's separated from osseous tissue by the endosteum of the bone. Blood moves into the bone marrow tissue through small arteries that come in through the nutrient foramina on the bone surface. And they'll penetrate the bone and empty into these large sinusoid spaces. Blood flows out of these sinusoid spaces in the same way that it came in, but in the veins. The sinusoids are lined by epithelial tissue like any other blood vessel, and they're surrounded by reticular cells here and reticular fibers. The spaces between the sinusoids are filled by cords or islands of hemopoietic cells. In them, we'll find macrophages and blood cells in all different stages of development. As the blood cells mature, they'll push their way through the reticular and endothelial cells out into the sinusoids and then flow away in the bloodstream. Now the thymus, which is our other primary lymphatic organ, is not only a member of the lymphatic system, but a member of the endocrine and immune systems also. The thymus houses developing T lymphocytes, as well as secreting hormones that regulate their activity as they mature. It's a bilobed organ that you can see here, and it's located in the superior mediastinum between the sternum and the aortic arch. We see that the thymus degenerates or involutes with age. That is because young children are much more active in developing their immune functions than we are as we age. The thymus is divided into several different lobes by the formation of trabeculae given off by the fibrous capsules. These trabeculae, or septa, divide up the gland. You can see them clearly here in this micrograph. We have different lobules separated by trabeculae. Each lobe has a cortex and a medulla. You can see here the cortex, the outer portion, and here the medulla. Both portions are populated by T lymphocytes. The cortex is populated by immature T lymphocytes. You can see those represented as purple dots here. The cortex is separated from the medulla by these reticular epithelial cells. Remember, these branched type cells that create little compartments. And in the cortex, they seal off the space so that the T lymphocytes can't go anywhere. As the T lymphocytes mature, they move towards the medulla. In the medulla, the reticular epithelial cells allow the T lymphocytes that have become immunocompetent to leave the thymus. This tight bonding of the reticular epithelial cells in the cortex produces the blood-thymus barrier. So there's a barrier where no blood is going to mix with the T lymphocytes that are in the cortex. The same barrier does not exist in the medulla portions of the thymus. Besides forming the blood-thymus barrier, these reticular epithelial cells will also secrete hormones and signaling molecules that promote the development and action of the T cells. They secrete such things as thymosin, thymopoietin, thymulin, interleukins, and interferon. If the thymus is removed in a newborn mammal, none of the immune functions develop, and the other lymphatic organs seem to depend on the thymosins or the T cells. So if they're not present, they don't develop properly either. Now on to the lymph nodes. Lymph nodes are the most numerous of lymphatic organs. There are about 450 in a young adult. And all these lymph nodes serve two primary functions. One is to cleanse the lymph, and the second is to act as a site for T cell and B cell activation should there be any invaders. Lymph nodes have sort of an elongated bean-shaped structure, often with a hilum, this area right here, an indentation on one side. It's enclosed in a fibrous capsule that also has trabeculae that divide the interior into compartments. The stroma, or the connective tissue of the lymph node, is primarily composed of reticular fibers and reticular cells. The parenchyma, or the functional portion, is divided into cortex and medulla, the cortical region being this outer portion here, and the medulla region being here. The cortex is then further subdivided into nodules. Remember, nodules were dense masses of macrophages and lymphocytes. The nodules of the cortex are where B cells multiply and differentiate into plasma cells when the lymph node is fighting a pathogen. Now, lymph enters these nodules through afferent lymphatic vessels. The lymph then moves into the subcapsular space and then moves through the sinuses of the cortex and the medulla and then flows out through one of the efferent lymphatic vessels that leads through the hilum. The lymph nodes act to really slow down the lymph as it's traveling through and clean it quite thoroughly. In fact, it becomes about 99% clean as it flows through a lymph node. Now, as lymph flows through one node after the other, it ends up being returned to the heart. almost free of anything that shouldn't be there. The lymph nodes have several classic locations. There are cervical lymph nodes. The cervical lymph nodes are the ones we see in our neck. There are a deep and a superficial group. These monitor the lymph coming from the head and neck and cleanse it. The axillary lymph nodes we've already taken a brief look at. They're concentrated in the armpit and they receive lymph from the upper limb as well as from the breast tissue. The thoracic lymph nodes are in the thoracic cavity. They're especially embedded in the mediastinum. They receive lymph from the mediastinum, the lungs, and the airway, and can intercept any pathogens as they enter at that point. There are abdominal lymph nodes. They occur in the posterior abdominopelvic wall. They monitor lymph from the urinary and reproductive systems. We'll see mesenteric and intestinal lymph nodes. They're found on the mesenteries that support all of the intestines and adjacent to the appendix. They monitor lymph from the digestive tract. There are also inguinal lymph nodes in the groin. They receive lymph from the entire lower limb. And then finally, there are popliteal lymph nodes. They're located on the back of the knee, and they receive lymph from the leg proper, or the lower leg. You can see here the arrangement of mesenteric lymph nodes that are associated with the large intestine. Each of them purifying a portion of the blood that's coming back from the intestines, as well as checking it for any pathogens. When a lymph node is under challenge from a foreign antigen, it may become swollen and painful to the touch. This is a condition called lymphadenitis. Physicians will often palpate the lymph nodes for lymphadenitis to see if there's potential of invading pathogens. So how are lymph nodes really involved in metastasis of cancer? Metastasis is a phenomenon where cancer cells break free from the original or primary tumor and travel to other sites in the body where they'll establish new tumors. As they metastasize, they could enter the lymphatic vessels. If they do this, they tend to get lodged in the first lymph node that they encounter. They can multiply there and eventually destroy the node. They tend to spread to the next node downstream and so on and so forth. But if we can catch them early by removing the lymph node most directly close to the tumor, then we could prevent them spreading anywhere else in the body. This is how breast cancer is often treated. First, by lumpectomy or mastectomy, along with removal of the nearby axillary nodes. Now we move on to look at the tonsils. Tonsils are patches of lymphatic tissue that are located at the entrance to the pharynx. They guard against ingested and inhaled pathogens. Each tonsil is covered by an epithelium, which invaginates to form these tonsillar crypts. The tonsillar crypts often will have food residue or lymphocytes or bacteria caught in them. Tonsillitis is when the tonsils become inflamed because they're actively involved in the removal of pathogens. Sometimes we'll undergo tonsillectomy to remove the tonsils if they continue to present the same problem. There are three main sets of tonsils. The palatine tonsils are right here on the posterior margin of the oral cavity. These are the ones that are most often infected as they protect us against both inhaled and consumed pathogens. The phalangeal tonsil is a single tonsil back here on the wall of the nasopharynx, otherwise known as adenoid tonsils. And the lingual tonsils are located here at the root of the tongue, one on each side. So the spleen is the largest of our lymphatic organs. The parenchyma, or the functional tissue of the spleen, exhibits two types. There's red pulp that you can see here and white pulp here. The red pulp results from sinuses that are filled with erythrocytes that seep out of the capillaries into these splenic sinuses. The white pulp consists predominantly of lymphocytes, a macrophages. You can see that it surrounds a small branch of a splenic artery here. It surrounds this branch like a sleeve. So although it looks like an ovoid or eggy type mass in here, remember this is a three-dimensional structure where the central artery would be coming out of the slide towards you, and it would be sleeved by this white pulp. The spleen has several different functions. Its primary function in the fetus is the production of blood. We lose that function in adults, except in the case of extreme anemia. It also serves as a blood reservoir. As these red blood cells seep out into the red pulp region, they're stored here. Now, the vessels in the spleen can get very, very small. So red blood cells, as they age and have to bend to move through these, may disintegrate, at which point they'll be phagocytosed by the macrophages. So it serves also as an erythrocyte graveyard. The white pulp monitors the blood for foreign antigens and will activate the immune system if invading antigens are found. You can see where the spleen is located here in the abdominal cavity, in the upper left quadrant. It's a highly vascular organ, and thus it's very vulnerable to trauma as well as infection. In the case where people have a ruptured spleen, it's so difficult to fix, they'll often perform a splenectomy and remove it. Although the spleen has a big role in immune function, an individual can live without a spleen. However, they'll be a little more susceptible to infections. So this concludes our section on the lymphatic system. We've certainly covered a lot of information. Please don't forget to go out to Anatomy and Physiology Revealed and review the materials there. And at this moment, you might close your book and note down each of the lymphatic organs and some of the general characteristics of them. Perhaps put together a little diagram, a picture of each, so that you get a good idea of the relationship of the different tissues within them. Also review the lymphatic tissues. We had two types, the lymphoid tissues and then lymphatic nodules. So we start out simple with diffuse lymphatic tissue, and then it becomes more organized in the form of nodules. And then even more organized with the formation of capsules, we see the lymphatic organs. Take some time here to review this material, and especially watch any animations that are available in Anatomy and Physiology Revealed. We'll see you soon for the next section, where we'll get into non-specific resistance.